We're live, huh? All right. Well, welcome to the School of Marine Science and Ocean Engineering Graduate Research Symposium 2021 version. I'm Eric Chapman. I'm Director of New Hampshire Sea Grant and Associate Director of Outreach for the Marine School. And today we have the honor to showcase graduate students' work from across campus focusing on marine topics. And as we usually do, we have virtually equal parts marine biology, ocean engineering, and oceanography with lots of interconnected combinations of these subdisciplines and more subdisciplines. That's one of the things that makes that's special about UNH and what we're able to do here. We have expertise across disciplines, we're connected, and we're all doing really important work that's desperately needed by society as we attempt to improve our relationship to marine and coastal resources. Today's focus is on those graduate students. They are in many ways the leaders of our community. Their passion, their curiosity, their willingness, and in many cases, their insistence on pushing boundaries and focusing on making a difference affects us all and makes us all better at what we do. This symposium highlights the work of students who are approaching the end of their graduate careers, and it gives them a chance to share their scientific stories with their peers, colleagues, and their mentors, and the broader community. The students here are on the launching pad of their careers, and those who participate go on to great things, but they don't go far, they stay involved. And today we're lucky enough to have a couple previous winners of our best oral presentation at the symposium returning to support the event. We've got Liz Widener, who was part of the the steering committee for today's symposium, and she's helping to moderate today's event. And then we have John Taylor, JT, who I had in my intro to marine biology class way back when, and he's gone on to do great things as a grad student, and now he's piping in from Sweden to participate as a judge for the oral presentation award. So thanks for coming and celebrating the UNH Marine and Coastal graduate students. And before we move on, I want to quickly thank the steering committee, and especially Sally Nelson, for making it so much fun to plan the event and for doing such an amazing job considering all the options and challenges of the symposium, our second one during COVID times. So thanks, Sally, Tara Hicks Johnson, Kaiser Vogel, Jessica Hinckley. And before we move on, I want to also share a land, water, and life acknowledgement. As we all walk the trail to life, we wish to acknowledge the spiritual and physical connection the Pennacook, Abenaki, and Wabanaki peoples have maintained on Indokina and the Aki land, Nibi, water, Lola, Queequeg, flora, and Awasak fauna, which the University of New Hampshire community is honored to steward today. We acknowledge the hardships they continue to endure after the loss of unceded homelands and champion the university's responsibility to foster relationships and opportunities that strengthen the well-being of the indigenous people who carry forward the traditions of their ancestors. Here's the agenda for today. We've got 4 speakers in the morning. We'll have a bit of an extended lunch break, and then we'll follow up with 4 more speakers in the afternoon. And with that, I'd like to hand it over to the director of the Marine School, Diane Foster, who would like to share a few words with you.
Thanks, Eric. I'd like to say welcome to our students foremost. This represents the best of who you are and what you do and why you came to UNH. I'd also like to say welcome to our faculty and staff and guests, and a special note to our UNH community. This is not the year that any one of us would have chosen, and we've all had to learn to work in new ways, and we've had to learn how to work together while being apart. And it's so impressive to see the hurdles that people have overcome to carry on. And we know that we're all desperately looking forward to getting back to campus in a way where we can see each other in the hallway and we can have a cup of tea, or we can meet at the pier to think about science or to connect with one another. We know that that day is coming, and we're really looking forward to that moment as we move through this time. And and yet I think it's really important to recognize what everybody has accomplished in this year. And it's been a tremendous amount. And you're continuing to address just critical questions that involve anywhere from our local coasts and our local waterways to our global oceans. And some of you take that from a exploration perspective where you're choosing to go or ask questions where nobody has gone before. Some of you are really focused and honed in on addressing really critical science questions that we really need to address tomorrow's solutions for how we feed the planet, for how we create energy, for how we reduce our dependence on fossil fuels, for how we interact with waters, for how our communities will live. So for that, I guess I say a hearty thank you. It means a lot to me. It means a lot to our entire university community. And I think if you look at the population that has joined us today, you'll recognize that the work that our students are doing is important to just a wide array of people. And we have people calling in from various places of the planet, across the planet. So I want to talk just a second about community and sort of how we're giving people things to look forward to. We have a number of new initiatives that we want to remind people of. Our business and proposal seed grants. Those calls have been out. We also remember that we have a call out for our graduate student research funding. And through our recent success with our 603 challenge, we raised about another $12,000 that will all go to support graduate student research projects. Those are projects that are pitched by grad students, performed by grad students, and it basically is one of our ways that the community says that we value what you do. We also have a number of other things. Our vessel naming competition is wrapping up, and you'll see the results of that soon. So we have a new 33-foot vessel that was provided by a very generous gift to help us develop capacity in the area of sustainable seafoods. And that vessel will be available for our broad UNH community. And in order for many of us to sort of better learn how to work with our small boats, Dave Shea, our Associate Director of Facilities, is actually going to be launching a small boat class that will be primarily directed towards our graduate students. Faculty know that you'll have lower priority than the grad students. And we look forward to more information about that. We anticipate that program to come in July, and we think that it will serve multiple missions. One is it'll help bring our community together, and it will also help develop skills. And with that, please look forward to new ways we can reconnect. Please find ways of meeting people outdoors so we can start to reinvest in our community. And now I'll give the floor back to Eric. Eric, and I guess I need to say thank you to the Outreach Committee. There's so much of the work that they do is significant as they're reaching out to students and faculty, and they're pulling together a number of ideas and then actually having to execute them. And so many people here have contributed to that. And to those people, I say a personal thank you. Thanks, Eric.
Awesome. Thank you, Diane. Really exciting stuff. Oops, this is not session 2. I apologize. Let me stop my sharing. I still have some Zoom kerfluffles that I fall into every now and again. Our first speaker is Alexandra Padilla. She is an ocean engineering PhD student who should be finishing up this fall. Very exciting. Her focus is on underwater acoustics, scattering from bubbles. So working to improve our understanding of gas release from natural hydrocarbon seeps, bubbles of methane and other gases. And this is important because methane is a potent greenhouse gas, has been implicated in global warming, and these seeps also support really interesting and productive benthic communities. Alexandra's advisor is Tom Weber, and she's going to be talking about acoustic monitoring and tracking of natural hydrocarbon gas bubbles over the course of a year. Take it away, Alexandra.
Thank you, Eric. Can everyone see my screen? All right. Thank you, Eric, again for that introduction. And thank you all for being here today. Before I begin, I want to acknowledge that the research presented today was done on Native Indigenous land, both in New Hampshire and in Santa Barbara, California. We acknowledge with honor and gratitude the land itself and the people who have stewarded it throughout the generations. And finally, before I start the talk, I really want to acknowledge the different agencies that funded this research and the many people involved throughout the process. And so with that, we can move to the West Coast to a place called the Coal Oil Point Seep Field. The Coal Oil Point Seep Field is a well-known natural hydrocarbon seep site, and it's known for its prolific hydrocarbon activity and has been studied for many decades. Researchers have observed both temporal and spatial variability and gas flow and gas flux estimates in the area and have hypothesized that this variability is linked to external physical processes such as tides or even due to offshore oil production from oil rigs. And so in recent years, an oil platform within the Coal Oil Point Seep Field, known as Platform Holly, has become inactive and decommissioned, and there has been a resurgence in the natural hydrocarbon seepage activity in the vicinity of the platform based off of anecdotal observations. And so this led a group of us from UNH and UCSB to map the spatial distribution of the seepage activity in the Coal Oil Point Seep Field. And during the study, we were able to identify a large patch of high seepage activity near Platform Holly. And so this shut-in at Platform Holly therefore provided us with the opportunity to deploy a long-term acoustic monitoring system to study the spatial and temporal variability of gas flux in the region and to see how it can be affected by external physical processes. And so to do the work that I'm going to talk about today, we mounted a Simrad ES200 split beam transducer to one of the cross members of the platform facing towards the westward direction where there's high evidence of acoustic seepage activity. And this transducer was also mounted at a declination angle of 15 degrees facing towards the seafloor. And so this transducer collects acoustic scattering measurements of targets within the acoustic beam, in our case, mostly gas bubbles. And the split beam capability of the transducer allows us to track the position of these gas bubbles in the acoustic beam of the transducer itself. And so this transducer was configured to transmit an acoustic signal every 10 seconds and has been collecting data since early 2019. So therefore, that means we have more than a year's worth of acoustic data that needs to be analyzed and processed, which is highlighted in this compilation of acoustic echograms for each of the months that we have. On the y-axis, we have the range from the transducer face. On the x-axis, we have the date. And then the color bar represents the target strength, where target strength represents how strong bubbles are scattering sound in the acoustic beam. So warm and cool colors represent low and high and low target strengths respectively. Between 10 and 130 meters, we have the acoustic water column data where we observe scattering from the gas bubbles that exist in the field. And beyond 150 meters, we have scattering from the seafloor. And so if we focus on the water column data between the 10 and 130 meters, we can immediately observe both temporal and spatial variability in the location and intensity of the acoustic measurements. But really what we're interested in understanding is in temporal variability in the estimates of gas flux and flow itself. So we really need to find a way to relate our acoustic measurements to these gas flux and flow estimates. And so what we need to do, what I'm going to discuss is how we use our acoustic data and the split beam capability of the transducer to infer information about different plume structures that exist in the data set, and that will help provide more insight on how to make this conversion between acoustic measurements to gas flux and flow estimates. And so what I'm going to do is first highlight a few of these bubble plume structures, because what we're going to do is once we've identified these structures, we can talk about governing equations that will relate our measurements. And so what I have here is our acoustic echogram, but also now we're going to look at a longship and athwartship angle data. So this is the position of the bubbles in the acoustic beam. And this is obtained through split beam data processing. And both of these angles are given in degrees. The bottom diagram here represents a simple cross-section view of the acoustic beam, where the longship angle is roughly in the up and down direction, and the athwartship angle roughly the left and right. And so the first example that I want to discuss is when in one direction, in our case, the athwartship angle has a single constant angle value, while in the other direction, in this case, the longship, we have this linear ramp up of angle from low to high angle values. And so if we were to think about what this may look like in the acoustic beam or in nature, this type of angle structure corresponds to a discrete bubble column intersecting at different locations in the beam as it rises through the water column. If you were to extend this discrete bubble column into a row of discrete bubble columns, one of our angles would remain constant while the other would become random and incoherent, which is a sign of a linear bubble plume feature, in our case, a bubble screen. And finally, if we were to extend this bubble screen over some area, now we have that both of our angles become random and incoherent, which corresponds to the case of a diffuse bubble cloud. And so now that we've identified our three major bubble plume structures, we need a way to easily detect and classify these structures in an automatic way so that we can process the year's worth of data that we have. And so to do this, we use a parameter known as the coherence factor, which measures the coherency between two different variables. In our case, we're interested in calculating the coherence factor in the alongship and the athwartship directions, which are shown in these plots here at the bottom of the screen. And so if we establish some threshold value, in our case, it's given by the lower limit of each of the color bars. If our estimate of the coherence factor is larger than said threshold, then the bubble plume structure is coherent, which means that it is well localized in the acoustic beam and we can determine its location. If the coherence factor, however, is smaller than the threshold, then the bubble plume structure is incoherent, which means that it's well distributed within the acoustic beam and therefore we can't determine its location. And so what this provides us with is 4 different ways to classify our bubble plume structures based off of their estimated coherence factor. If the coherence factor is coherent in both directions, we are observing a discrete bubble column. If the coherence factor is coherent in one direction, but incoherent in the other, then it's a bubble screen. And finally, if the coherence factor is incoherent in both directions, then it is a bubble cloud. And so with these classifications, we can generate an acoustic classification map where each acoustic sample is classified as either a bubble column, a bubble screen, or as a bubble cloud. And so now that we can classify these different bubble plume structures using the acoustic data set alone, we can discuss some of the mathematical equations that we can use and that are needed to convert our estimates of acoustic scattering to estimates of gas flux and flow. And so to start, we're just gonna talk about the simplest case, which is the discrete bubble column, which means that it is coherent and localized in both the alongship and the athwartship directions. And so for a discrete bubble column, we can calculate the gas flow and flux given this set of equations here, where the gas flow is calculated by integrating the product of the volume of the gas bubbles by their rise velocity over a given size bubble size distribution, and multiplying that by the number of bubbles per unit height. And then the gas flux is simply our gas flow normalized by some unit area. Now, when we think about our acoustic estimates or the acoustic measurements that we're obtaining from our data set, what we're looking at is something that's called the total backscattering cross-section, which is just the total scattering from all of the gas bubbles that are in the acoustic beam itself. And it's given by this equation where we have the number of bubbles per unit height multiplied by the vertical extent of the plume that intersects the acoustic beam, and then is multiplied by the average backscattering cross-section for a given size bubble distribution. And so what we can immediately see is that there's a common term between our equation for the total backscattering cross-section and also the gas flow and gas flux equations. And what this allows us to do is to obtain a set of equations that directly relate our acoustic measurements to gas flux and gas flow. And so we can extend these set of equations for all different bubble plume structure cases. However, these equations become a little bit more complicated as we look at different bubble plume structure types. So if anyone would like to talk about this offline, I'd be really happy to do so. But the main takeaway is that now we have a direct path to use acoustic data to identify different bubble plume structures and to take those acoustic measurements into estimates of gas flux and flow for all of these different bubble plume structure types. And what this will finally allow us to do is to obtain some long year time series of gas flux and flow that will provide insight on the long-term variability of gas flux and flow in the region. So is this gas flux increasing with time, decreasing, or is it staying constant? But what it also enables us to do is to correlate this resulting time series of gas flux and flow with the time series from external physical processes such as tides and atmospheric pressure, and determine if these external processes affect gas flow and gas flux estimates in the region. And while we mostly just talked about a particular seep near platform Hawley, we can extend this study to other natural hydrocarbon seep sites that exist across the ocean seafloor and potentially start to address questions about global estimates of gas flux and flow from natural hydrocarbon seep sites and lower the uncertainty in these estimates that currently exist. And so with that, I'm ready for any questions.
Thank you so much, Alex. That was a really wonderful presentation. If anyone has any questions for Alex, you can feel free to unmute yourself. We have time for a couple of questions, or you can type them in the chat and I will read them out loud. So while everyone's thinking, I have a question, Alex. So when you used the coherence factor to classify your various seep geometries, I guess, for the bubble cloud, you said that you had incoherency in both of your two variables, but wouldn't you also have incoherency if there were no bubbles in your beam, like if you were insonifying empty water column space? How do you deal with that?
Yeah, that's a great question. And yeah, you would expect to see incoherency if you had nothing in the water column. And what we usually do is, so here we have the acoustic echogram, and all this black space that exists is what we would call noise. So what we would do is filter out the noise based off some, target strength threshold. So if they're below this threshold, then it's probably empty water column noise. And that's how we would distinguish between gas bubbles or targets versus noise.
Looks like Diane Foster has a question.
I have a question, Alex. So in my limited understanding, I believe that bubbles change size and rise rate as they move through the water column. And yet you've defined thresholds for looking at these three different threshold conditions. How sensitive are you to the bubble sizes, particularly as it rises through the water column?
So that would be, we wouldn't, I guess, just to make sure that I understand what you're asking, we can, if the bubble size is changing, if they're still in a particular configuration, then that shouldn't affect us identifying what kind of bubble plume structures we're looking at, but it would affect how we would measure gas flow and our target strength, because it depends on that bubble size distribution and how the rise velocity might change. And so we would need to potentially run some models to understand how at different depths what the bubble size distribution would be, to take that into account, I think.
And do you see that they change? Do you ever see evidence as it goes through the water column that you go from one type to another type?
Of different plume structures?
From a single to a screen?
Yeah. We do, yeah.
Thanks.
Yep.
All right, so we'll have to wrap it up there. I note there's a couple of questions in the chat, and we're going to have time after all four presenters in this first session present for everyone to ask them questions in individual breakout rooms. So Ben, hold on to that question and you can ask Alex afterwards. And now Eric will introduce our next speaker.
Thank you, Liz. Now you see what I mean when I say these are the leaders of our community. Incredible talk, Alexandra, and a great presentation. It's just a reminder that we all have to raise our game with communicating science and using PowerPoints. That was unbelievable. Thank you. Our next speaker is Grant Mccown, who is focusing his work in marine biology and finishing his master's this spring. Greg Moore is his advisor, and his focus generally is in marsh ecology and restoration ecology. And this is really important. His work that he'll be talking about is focusing on living shorelines. And this is critical to establishing buffers in our natural systems that builds the resiliency of our coasts to things like sea level rise, increasing frequency and intensity of storms, and other climate effects. The topic of his talk today is short-term restoration performance of living shorelines in the Great Bay Estuary of New Hampshire. Take it away, Grant.
Awesome. Can everyone hear me? And did I do the screen sharing right? I'm still getting used to two screens. Awesome.
Good.
So thank you so much for the introduction, Eric, and thank you to all those in the SMSOE community for allowing me to speak today. I'm really excited to present what I've been working on these past few years. Just a note, if you are using Zoom in full screen, I've devised this presentation so this little Zoom window should go in the top right-hand corner as to not cover up any photos or text. I would also just like to take a few seconds to all those who have helped me in the field, especially my committee, those who have funded me, and those who have helped me in the field in the lab. So, over the past several decades, researchers, ecologists, and coastal planners have been calling for the use of nature-based methods to stabilize shorelines and promote the health of our nation's estuaries. And one of the main methods that has been especially promising is called living shorelines. Living shorelines is the combination of traditional shoreline engineering, such as seawalls, and ecological restoration of coastal habitats. Now, one of the most common techniques for living shorelines is the creation of a salt marsh and a protective cove, plus the construction of a 3 to 4 foot riprap sill. You can see in the bottom right-hand corner. The thinking is that this riprap sill will initially reduce wave energy and provide time for the vegetation to root out and stabilize the soil, while over time, valuable ecosystem services and the resilience of the shoreline will increase as that salt marsh recovers. An example of a living shoreline is one of my study sites at Wagon Hill Farm in Durham, which I'm sure many of you are familiar with. The site underwent rapid shoreline erosion, but was corrected with this classic living shoreline design. So why am I even talking about living shorelines? Well, over the past 5 to 6 years, the New Hampshire Department of Environmental Services has been steadily shifting their focus to living shorelines as their regulatory preferred shoreline stabilization method. In 2019, the title Shoreline Stabilization Permit was changed to give preference to living shorelines. Additionally, the state has directed funding into 3 pilot projects since 2016 in the Great Bay system. However, despite this growing emphasis on living shorelines, it's still a relatively new science with only a handful of published studies and reports on their efficacy. Additionally, almost all of these published studies and reports have been located in the Chesapeake Bay and the North Carolina coast. And this lack of knowledge on the performance of living shorelines, especially in a northern New England context with ice rafting and shorter growing seasons, may leave researchers and regulators flying blind in terms of expectations and maintenance needs. So I set out to improve, so in order to improve the outcomes of future projects, I set out with 2 research objectives. First, I quantified the short-term restoration performance of these projects. And second, I identified adaptive management needs within the first 5 years. So to carry out this research, I conducted monitoring at those 3 pilot projects, North Mill Pond in Portsmouth, Cuts Cove in Portsmouth, and Wagon Hill Farm in Durham. And to quantify the recovery of these salt marsh systems, I designed a modified before-after control impact experimental design at each site, where each living shoreline was paired with a local reference salt marsh and a degraded shoreline. To gauge the restoration performance, I monitored 3 aspects of the salt marsh system: habitat structure, fauna use, and biogeochemical processes. For habitat structure, I conducted plot sampling of the vegetation to measure visual cover and plot level species richness. For fauna, I conducted 1 round of minnow trapping at each site to monitor mummichug abundance and adult length. And for biogeochemical processes, I collected pore water at each of the vegetation plots for salinity, redox potential, pH, and sulfides. All sampling was conducted in August and September of 2019 and 2020, while minnow trapping only took place in 2020. So to quantify the recovery of these living shorelines, I deployed the Restoration Performance Index, which is an evaluation tool created by my advisor, Greg Moore. The RPI is an evaluation tool that measures both the progress achieved of the living shorelines based off that degraded shoreline and the progress remaining based off that reference salt marsh. In order to read the RPI, it provides a score from 0 to 1, where 1 means that the living shoreline has reached parity with the reference salt marsh conditions, i.e. it's reached the restoration goals for the project. Additionally, I applied logistic regressions onto these RPI scores against project age to get a better picture of the trajectory of this recovery. And to better understand the RPI scores themselves, I conducted student t-test comparisons for certain metrics between the living shoreline and the reference. So after 2 years of monitoring and being in the marshes, the living shorelines displayed a pattern of rapid recovery, which was primarily driven by the vegetation seen in green and the nectin community seen in orange. I should note that in years 0 and years 3 on the graph were samples in 2019 and do not have any nectin data. Additionally, the biogeochemical processes shown in blue continue to lag early on in the recovery. One way to read this graph is that basically a little over 50% of ecosystem structure, function, and services recovered within 4 years at these living shorelines. On the right, we had a well-fit 3-parameter logistic regression solidifying the trends seen on the left. The logistic regression identified a plateau of recovery, fitting at roughly 2 years after construction at an RPI score of 0.52, recognizing that there's now a new phase of recovery that's more slow and incremental. I do want to point out that that Wagon Hill Farm project I've been mentioning recovered just surprisingly quickly within 1 year. It increased from a score of 0.04 to a score of 0.51 within 1 year after construction. When diving deeper into the RPI scores, we find that the vegetation score followed a similar shape trajectory with the platform at roughly a score of 0.67. In other words, the vegetation community reached roughly 70% of the structure and species richness compared to the reference salt marsh before it slowed down. And when breaking it down to the visual cover of the vegetation community only required 4 years to reach statistical equivalence with the reference salt marsh. In response to the mummichugs, the community recovered only within 1 year. Mummichugs responded really quickly to the presence of new salt marsh habitat and immediately began utilizing the space for refugia and feeding. However, the soil conditions of the living shorelines remain significantly more aerobic compared to the highly anaerobic conditions of the reference salt marshes. Living shorelines are constructed with highly porous sandy fills, which allows for rapid drainage after each tidal cycle. And if you think about in the long term, this prevents the accumulation of organic matter and the development of those anaerobic conditions, which might require much longer timeframes in the vegetation and the fish community. In addition to the restoration trajectories, I compiled 3 major adaptive management needs. First, we experienced intense geese herbivory from October to December in the Cuts Cove living shoreline in both years. Now, geese are known to eat rhizomes and the roots of Spartina. We've observed it in other marshes. However, this sandy fill allows these geese to basically essentially rip out entire plants and just send this whole project back a few years. We witnessed large damages, and this site will recover additional plantings. The second issue we identified while monitoring was just a high mortality of Spartina plugs within the first few meters of the seaward edge, so right behind that sill. We experienced this both at North Mill Pond and Cuts Cove. For example, at North Mill Pond, close to 50% of the original planted low marsh, which is completely bare within 4 years. And these areas will just require more planting to really establish that vegetation. The third possible issue at living shorelines is high raft deposition that smothers and just kills off the vegetation community in the high marsh. We saw this at North Mill Pond and a limited degree at Wagon Hill Farm. To prevent this vegetation die-off, rack will need to be raked up and removed after pulse events like storms or just seasonal deposition. So the management implications for this research can be broken down into 2 components: 1, expectation for future projects, and 2, maintenance requirements. For future expectations, practitioners should expect 2 phases of recovery, with the first being a rapid recovery driven by vegetation and nectin, and then a second phase of slower, more incremental recovery of wetland soils. This rapid recovery should translate into roughly 50% of ecosystem structure and services compared to the reference salt marshes within a few years. And we saw Wagon Hill Farm achieve that within just 1 year. Secondly, during the planning phase of these projects, stakeholders should include monitoring and maintenance costs into their budgets. We identified 3 maintenance needs, which will be annual plantings for the first 5 years, proactive beast management efforts, and raft removal. So at the end of the day, living shorelines show incredible promise in stabilizing eroding shorelines, as well as restoring valuable coastal habitats and ecosystem services in the Great Bay Estuary, and should be continued to be utilized. And with that, I'll take any questions.
Wow, Grant, that was a fantastic talk. If anybody has any questions for Grant, I see Nathan has a question, so I will turn it over to him.
Thank you, Grant. I really liked the slide organization. It was very easy to follow, very clear, very crisp. I do have a question. This might not be essential to your research, but when you were doing those, I don't know if they were logistic fits, the sigmoid, those S-shaped curves. I guess two questions.
Yeah.
One, how sensitive are those to outliers? So if you were to move the year 0 point, what would be your fit? And two, do you think that a 1-year sampling interval is sufficient to characterize the changes in the first year and a half or so?
Yeah, so this was my first really good run at nonlinear modeling. So I would assume that outliers would probably affect this. The gray bars here represent just the standard error, so it's not a 95% confidence interval, but I would assume the outliers would probably have an effect. I was surprised at how well this did with an R squared of 0.94. To answer your question on the monitoring aspect, we follow typical monitoring guidelines for restoration projects, which is just 1-year intervals. So we try just to follow normal, I guess, industry standards for these. The reason we did it in August and September is that's when the vegetation has its usually greatest biomass and canopy height and full visual cover. Hopefully that answered your second question.
Yeah, just for the sake of time. Yeah, thank you.
Yeah, definitely the minnow trapping should be done more active, and that was more of a kind of a last minute thing. So I would definitely think the minnow trapping should be done more frequently throughout the summer.
Okay.
We have time for a quick question, so we'll take Peter.
Hey, Grant. Are there any natural analogs to the sills up here in New Hampshire, New England? You know, kind of analogous. I spent a lot of time in North Carolina, so oyster reefs down there kind of look familiar.
Yeah, so the analogy or the analogous habitat that always comes up are oyster reefs in terms of this 3-dimensional hard structure that things can colonize. Another piece of my research, which I didn't have time to talk about today, is we did monitor the algae colonization and the invertebrate colonization of the riprap sills. We found that algae like fucus and ascophyllum really started to colonize them and got up to 50% within a few years. And we saw barnacles starting to colonize the area too. But we haven't seen any like ribbed mussels yet. But a lot of people hypothesize that these riprap sills are great for habitat heterogeneity and will probably provide even more refugia for the nectin community as well. That's seen in the oyster reefs.
Grant, I see that we have a couple questions in the chat, but we're going to go ahead and hold off on those in the end, just so that we can introduce our next speaker. So those who have questions, please do talk to Grant after we finish all 4 presentations. Over to you, Eric.
All right, thanks. Great talk, Grant. And our next speaker is Jang Gwen Choi. He's a PhD student in oceanography, and Tom Lippman is his advisor. He focuses on ecosystem modeling driven by non-red field dynamics and will be looking at parameter sensitivity experiments. This is important because although many marine ecosystem models have been developed to simulate large-scale pelagic systems, they don't consider well benthic processes in non-red field dynamics, and that's dynamics that occur when the relatively stable ratios of nutrients in the ocean are no longer stable. And as a result of this, ocean models can't properly resolve coastal ecosystems in particular. So he's going to be talking today, focusing on expanded nutrient, phytoplankton, zooplankton, detritus models, considering non-red field dynamics and parameter sensitivity experiments. So take it away, Jang.
Hi, can you hear me? Okay, thank you for introducing me. So I'm Jang Choi. Feel free to just call me Jang. And today I wanted to talk about expanded nutrient phytoplankton to plankton detritus, which is called MPGD model considering non-random field dynamics. So first, we need to talk about what is MPGD model. MPGD model is one of the simplest marine ecosystem model, consists of a minimum number of variables. So these variables are linked to each other through prey-predation relation. So eventually, nutrient is uptaken by phytoplankton. Phytoplankton is grazed by zooplankton, and these planktons becomes detritus through their mortalities. Finally, detritus demineralized into nutrients. Eventually, they forms kind of a cycle of materials, of which currently is the nitrogen. So it must be noted that detritus is subject of vertical sinking. So this contributed to general ecosystem dynamics well known as marine snow. So however, they have some problem in the shallow region. In the deep pelagic region, detritus has sufficient time to be changed into nutrient before they reach the bottom boundaries. However, in the shallow region, they are quickly reach the bottom boundaries. Once they reach boundaries, they just keep sinking and just escape model domains before they change into the nutrient. Eventually, this process keep decreasing, continuously decreasing the nitrogen level in the systems. So because of this, eventually the model simulates unrealistic oligotrophic water mass at the shallow region. Usually we expect the productive water mass at the shallow region, something like this satellite image is shown. However, the model failed to resolve this productive water mass. This is not a problem of only simple MPGD model. These are the simulation results using Nemuro ecosystem model. Nemuro ecosystem model is one of the most complex marine ecosystem model. So you can see this complex model also failed to resolve productive water mass at the shallow region. So it should be also noted that this study using the parameter optimization. The fact that parameter optimization failed to fix this problem means that this is not a problem of choice of the parameters, rather than error from the model structure itself. Eventually, we need to fix the governing equation itself. So question is, how can we fix it? So here is our motivation. These are the phosphate and nitrate plot based on in situ measurements around the Korean Peninsula. So in the deep pelagic region, phosphate and nitrate shows a strong correlation, well known as Redfield ratio. This is what all of the ecosystem models are assuming. However, in the shallow region, so Redfield ratio does not work, and nitrate concentrations are frequently over or undersaturated than phosphorus concentration. This implies presence of additional source and sink of nitrogen, usually attributed to denitrification and nitrogen fixation. So this in situ observation clearly shows that we have to resolve these non-Redfield dynamics to describe coastal ecosystems. So now we should develop the model. So these are the combination of traditional MPGD model and schematics. I just recommend just focus on the schematics because these complex equations are nothing more than just mathematical description for those schematic. So first, we add phosphorus cycles. So we add a phosphorus cycle to explicitly describe non-Redfield dynamics. And then we specify the bottom boundary condition representing sediment denitrification, nitrification coupled processes. So this process describing that once detritus reaching the bottom boundaries, some portion of nitrogen is removed by denitrification, and the other portion is instantaneously changing into nutrient and then come back to water columns. So on the other hand, the phosphorus in the detritus is not subject to denitrification, so they are perfectly changing into nutrient form without any losses. So therefore, eventually, nitrogen in the system becomes dissipative. On the other hand, the phosphorus in the system becomes conservative. Finally, we add one more phytoplankton function group representing nitrogen fixers. So this phytoplankton functional group depends on only phosphorus. They does not require nitrogen for their growth. So eventually their growth assimilates organic matter including nitrogen without any consumption of the nitrogen in the system. So their growth is supplying additional nitrogen into the system, which corresponds with the nitrogen fixation. So this figure shows the simulation research using traditional MPGD model without Redfield ratio and expanded MPGD model with Redfield ratio dynamics. No, Redfield ratio dynamics. Similarly with the previous studies, traditional model without Redfield dynamics failed to resolve coastal productive water mass. However, consideration of these additional dynamics clearly revised the problem and successfully simulated productive water mass along the marginal coastal ocean. So now we should talk about how this expanded model fixed the problem and what is the internal dynamics of the model. Sorry, it is passed automatically. I don't know why. So this, with a little bit of assumptions, such as no zooplankton and simple parameterization for the data thinking, we can obtain steady state solution for the expanded ecosystem model. So eventually they are determined, the steady state phytoplankton concentrations are determined as function of model coefficient, especially function of depth itself. So here is the visualized steady state concentration of phytoplankton and written as a function of this non-dimensional number. This is indicating relative depth. So in the shallow region, both phytoplankton group coexist. In the mathematical models, in order for two different functional groups to coexist, they should be limited by different resources. Eventually, normal phytoplankton should be limited by nitrogen, and nitrogen fixers should be limited by phosphorus for their coexistence. So in the shallow region, there is intrinsically the dominant denitrification. So this suppresses growth of the normal phytoplankton through the nitrogen limitation. Eventually, nitrogen fixers becomes relatively competitive for phosphorus competition. So that means nitrogen fixers increase. That means the additional nitrogen will be supplied into the system, which will alleviate the nitrogen limitation of normal phytoplankton. Eventually, in the shallow region, there is negative feedback contributed to coexistence of the two phytoplankton. However, in the deep region, the nitrogen fixer concentration decrease and eventually converging to 0. So this is because in the deep region, denitrification becomes negligible and nitrogen becomes mostly conservative. So eventually, normal phytoplankton is not limited by nitrogen rather than phosphate. So in this case, two phytoplanktons competing for the same resource. In this case, they cannot coexist, and only one functional group can survive. That is which has better nutrient uptake. That is normal phytoplankton. So as a consequently, the nitrogen fixer in deep pelagic region is defeated by normal phytoplankton in phosphorus competition. So eventually, our steady state solution clearly shows that denitrification sustains nitrogen fixers by regulating phosphorus competition with normal phytoplankton. So it must be noted that this is not the first model resolving non-net field dynamics. There is a little bit complex model. They are already providing consideration of this kind of dynamics. However, we want to argue that this is the simplest model describing the coastal ecosystem, including the non-net field dynamics. So eventually, we highlight our model can be used for analytical studying, which can highlight the dynamics of the system itself. Yeah, thank you.
Thank you so much, Jang. That was a great presentation. I especially liked your model diagrams. Those were lovely.
Thank you very much. Yeah.
Does anybody have any questions for Jang? We have one in the chat from Diane Foster. Thanks, Jang. Can you discuss the threshold for shallow water?
Yes, definitely. So you can see this. here, this is non-dimensional number. Actually, H is water depth itself. WD divided by delta, that is relative depth. That is kind of a threshold of the depth. WD means this is the vertical sinking speed of the detritus. So it is kind of a meter per day. And delta is remineralized rate. So how fastly detritus changes into the nutrient. So it is dimension over 1 over time. So eventually we can WD over delta, that is the kind of threshold of the coastal water system.
I have just a short question. Does your model or the more complex models, do they incorporate the reintroduction of nitrogen from upwelling processes in coastal water?
Yes, definitely. Usually most ecosystem models are coupled with the hydrodynamics model. So as a complex model, definitely they do that. And this simulation results also from the coupled model. They are forced by realistic forcing, wind, tide, boundary currents from the boundary. They resolve most of everything, including rivers.
Cool, very nice. Thank you, Jang. If anyone has more questions for Jang, again, there'll be time after all the presentations are finished. So I'll turn it over to Eric for our last presenter of this session.
All right. Thanks, Jang. Great talk. Really important stuff. As a modeler in my conformal life, it's really exciting to see these important strides to improve the resolution of large-scale ecological modeling in the ocean. Important work in the background, very necessary for the future. So the last speaker of the morning session is Nathaniel Spada. He's finishing his master's this year with a focus on aquaculture, in particular on culturing lumpfish as a biological control for sea lice on finfish aquaculture operations. And this is part of efforts to develop ecologically sustainable approaches to aquaculture, which is a critical need to develop food security while removing some additional pressure on wild fisheries. Elizabeth Fairchild is his advisor, and his focus is on nutritional and density dependent effects on the growth of juvenile cultured lumpfish. Hand it over to Nathaniel.
Forgot to unmute myself. Sorry about that, guys. First, before I get started, I'd like to thank the School of Marine Sciences for the opportunity to present, and also to my fellow speakers. Your work is appreciated, and it's really cool. I'm loving the talk so far. So I'm also getting used to the two screen thing. So let me see if I can figure this out. All right, so thank you for the introduction, Eric. And today I'm going to be showing you guys some of my work on lumpfish, looking at the nutritional and density-dependent effects on the growth of juvenile lumpfish. So to paint the picture for you guys, I'm going to be talking about lumpfish and their role in salmonid aquaculture. So millions of tons of salmonids are produced annually, and the biggest economic woe that impacts this industry are these ectoparasitic copepods called sea lice. These infestations and these sea lice eat and rasp away at the mucous membrane and the flesh of these fish, creating these wounds and lesions that have a range of sublethal and lethal effects that effectively lower market value. In terms of mitigation, in the late 20th century, a lot of the mitigation methods were surrounded around chemical baths using chemotherapeutics. However, there was research found that these chemotherapeutics were having effects on non-target species. So a lot of countries banned the use of these chemicals. Growers are now looking for new mitigation methods, and one of which comes in the form of cleaner fish technology. Lumpfish act as cleaner fish when they eat the sea lice off of these salmonids in the salmon pens. Because of the usefulness of these lumpfish as cleaner fish, millions of juveniles are produced annually, with Norway leading the way at 43 million. This total is fast approaching 50 million juveniles produced per year. With all of these fish being produced, there's still not a single protocol that is drawn upon for growing these fish. So it leaves the question whether or not we're growing the lumpfish in the most economically efficient way possible. Also, something interesting to know is that the US production of lumpfish is pretty much solely based in UNH, which is kind of cool. This is getting into my thesis work. So I'm trying to find the best way to produce these lumpfish. And today I'm going to be presenting you two of the experiments that look to do just that. One of which being a diet experiment looking at how protein source and protein lipid concentration affect growth, survival, and fish aggression in these juvenile lumpfish. And another being a stocking density experiment looking at what stocking density maximizes growth and survival, but also limits fish aggression in these juvenile lumpfish. So lumpfish are cannibalistic. When you put them in these tanks, they like to nip at the caudal fins of their neighbors, So we wanted to quantify this fish aggression in terms of both the occurrence of which the fin nipping occurs, and also the severity of these fin nips, which I'll talk about in just a second. Some common methodologies between the experiments were growth was monitored bi-weekly by taking the wet weights of each individual fish. In terms of the fish aggression, we used this fin nipping scale. So each fish was given a score of 0 to 5, 0 being a perfectly intact caudal fin, and 5 being severe damage to the caudal peduncle. And we use this to get the severity score for each treatment. In terms of mortality, mortality was monitored daily, and any mortalities that were found were removed from the tank. Getting into the diet experiment, there were 8 diets tested. Diets 1 through 6 were made by the USDA and were our experimental diets. They were tested in quadruplicate, and diets 7 and 8 were our commercially available diets. So diet 7 is a biotrout diet, which is a salmonid feed that is fed to the salmon out in the net pens, and it's likely a diet that the lumpfish would be exposed to while in these sea cages. Diet 8 is a Spreading Europa diet, and it's a diet that we feed to the general population. These two commercial diets were tested in triplicate. So there were 15 fish stocked per tank at around 9 grams. The fish were fed at 3% body weight spread over 5 feedings per day. Something interesting to note is that diet 5 and diet 8 had the same protein lipid concentrations, but as it turned out, they performed differently from one another, which I'll show you in a second. Getting into the results of this experiment, there are only 2 mortalities found throughout, so survival was not impacted by these diets. Also, in terms of the occurrence of fin nipping between the treatments, there was no significance found there. But in terms of the severity of the fin nips, there was some significance found. But first, let's get into the growth data. Looking at this graph, this is showing the mean fish weights per biweekly weigh-in. We see that diets with higher protein concentrations at 55% are leading the pack. So diets 4 and 5 at 55.20 and 55.15 are leading the pack here. And diet 7 is showing the slowest growth. And there's a couple reasons for this. Diet 7, which is the biotrout, has the lowest protein concentration. Also, its protein source is plant protein rather than animal protein. Because of this so low growth in diet 7, or the fish fed diet 7, it was eliminated from analysis when looking at the overall percent growth from week 0 to week 10. And we see the same thing here. Diets with 55% protein showed the highest growth in the juveniles, and that lipid concentration did not have a significant effect on the growth of the fish. Again, remember when I talked about diet 5 and diet 8 having the same protein lipid concentration, it turns out that diet 5 outperformed, or the fish fed diet 5 outperformed the commercially available feed, which was quite interesting. And this is likely due to the ingredient differences between the two. Looking at the overall severity of fin nips between the diets, we see the same trend here. So diet 5 is again showing the least severe fin nipping when compared to the commercially available diet. Getting into the stocking density experiment, there were 4 densities tested, 40, 60, 70, and 90 grams per liter. The 40 gram per liter treatment is a density that is often used in hatcheries, so we thought we'd test that out. They were stocked in these 3-liter tanks and were tested in triplicate. The fish started out at around 13 grams and were fed 2% body weight spread over 3 feedings per day. And stocking density was maintained throughout the experiment, meaning that once stocking density was exceeded, some fish were removed from the tank in order to get down back to that baseline value. Getting into the results of the stocking density experiment, there were some surprising results. So no mortalities were found throughout the experiment. So survival was not impacted by these densities that were tested, but certainly as we get to more extreme values, this would likely not be the case. Also, in terms of the occurrence of fin nipping between the treatments, there was no significance found. But in terms of the severity of these fin nips, there was some significant trends, which I will show you in just a bit. Getting into the growth data, we see basically what we expect to see. So the 40 grams per liter is leading the pack, with the two intermediate values, 60 and 70 grams per liter in the middle, and the 90 gram per liter treatment showing the slowest growth. And we see the same thing when we look at the overall percent growth graph, with the fish in the 40 and 60 gram per liter treatment showing significantly higher growth than the fish in the 90 gram per liter treatment. And the 60 and 70 gram per liter treatment, the two intermediates performing similarly to one another. Looking at the overall severity of fin nips between the stocking densities, we see something pretty interesting. So we see that 70 gram per liter showed the most severe fin nips out of all of the treatments. And the reason for this is, as we increase in density, the social hierarchy forms amongst the fish. So the larger fish are going to start picking on these smaller fish and causing these more severe fin nips. But as we get too dense, there's just simply not enough room in these tanks for the fish to operate. These larger fish have less opportunity to cannibalize their neighbors, and therefore we see less severe fin nipping in this 90 gram per liter treatment. Some concluding remarks: diets made with animal protein and diets with high protein concentrations resulted in the greatest growth in these juvenile lumpfish. The stocking densities that we tested did not impact survival or aggression, but certainly these lower densities result in faster growth in the lumpfish. The importance of this research is we're trying to find the most economically efficient way possible to grow these fish. And without a single protocol that is drawn upon, it leaves the question whether or not we're actually doing this. Also, the story of the lumpfish is a sustainable one. So instead of using these chemotherapeutic chemical baths to treat the sea lice and affecting the ecosystem, we're using a biological method that is actually local in the Gulf of Maine. Also, lumpfish outgrow their usefulness. So when lumpfish reach a certain size threshold, they change diet preferences and therefore become less effective cleaner fish. So we can use some of these results to either speed up or slow down the growth of the juveniles in order to meet the needs of the salmonid facility. And with that, I'd like to thank the Northeastern Regional Aquaculture Center, the USDA, Memorial University, the University of New Hampshire, the New Hampshire Agriculture Experiment Station, the School of Marine Sciences, and also to my graduate and undergraduate colleagues, the Coastal Marine Lab Manager Nathan Reynolds, my colleagues at the USDA, and my graduate committee. And with that, is there any questions?
Thank you so much, Nate. That was really interesting. Loved all your pictures. Do we have any questions from the audience?
Hi, Nate.
Hello, let me see if I can get your face in here. Perfect.
Nate, wonderful talk. I agree with Liz. I always love seeing the lumpfish photos, and just really clean story and slides, and really job well done. I kind of want to, you mentioned this in your very last slide, but kind of thinking about, you find really specific trends with growth based on diet, but also densities, but also then animal welfare in terms of the aggression and fin nipping. So what are the next steps in terms of how do you then integrate this so that way the next folks who are using lumpfish and aquaculture can actually figure out what are those ideal combinations that you want to use to try to maximize the ability for those lumpfish to clean off the aquaculture fish while also maximizing the welfare of those animals?
Excellent question, Nathan. So it really does depend on the needs of the salmonid facility. So like I said, lumpfish outgrow their usefulness once they reach a certain size. So finding a rate and creating a combination is going to be facility. It's going to be dependent on the facility. Certainly, you'd want to maximize growth and increase the rate of growth, and to a point where you're doing the most doing it in the most economically efficient way possible. But like you said, animal welfare is important here. I would say that using the 40 gram per liter treatment would certainly increase growth. And we're trying to maximize nutrition as well with these diets. So I would imagine that finding the best nutritional content for the diet is most important. But feeding less of this diet would slow growth, or you could increase the rate of feeding for the diet to maybe produce larger fish if that's what you're looking for. It's really a gradient at this point.
Yeah, and do you think you'd be able to integrate across those experiments within some sort of like decision model or something like that based upon the facility and what they want to do? This is how many you should stock to be able to grow this fast and hold on to the fish for a certain time.
Yeah, and that's exactly what I'm trying to do now. I'm trying to replicate this data, and I'm doing another diet experiment. I did another stocking density experiment, trying to create, like you said, a model of sorts to do just that.
Awesome. Thank you, Nate.
No problem.
Lots of questions for Nate. I think we are at the end of our time for the first session. I believe we're going to be creating breakout rooms, but I will pass it over to Eric to sort of wrap everything up, and then everyone will have the opportunity to ask more questions of our presenters.
Shown here that will be a bit more understandable. And then we can ordinate each individual stomach here. So a point represents one stomach with its diverse community of prey items. And those closer together had more similar prey items, and those further apart were more dissimilar. When we overlay this with the time that stomach was collected, we can see different patterns between the 2 years for which we've done the stomach composition. And more closely, we also see a shift and a broadening of diets from the beginning to the end of this resource pulse. So their diets are flexible, and in particular, these changes are driven by a few associations to particular prey items that are more abundant at different times. So taking then this behavior of abundant but flexible feeding and attempting to look at that and transition into movement and determine how are sculpin moving and whether or not that is similarly seasonal. We use acoustic telemetry in order to monitor sculpin movement. And so acoustic receivers were positioned throughout Tremblay Sound, organized into a series of 7 gates with 36 total receivers. So those are shown in the red dots here, are receivers. And then fish were implanted with acoustic transmitters, so a total of 112 across the 3 study years. Those are implanted within the fish, and then as those emit a sound that can be detected by the receivers, we can monitor that fish as it moves. And this is done passively, so both in the ice-free season when they're released, as well as in the ice-covered season when we're not there, we can still monitor and track the fish as they move. And so as we do this, as a fish is detected at different gates and monitored over time, we can link those together into a sequence of their movement behaviors and start comparing fish that way. So when we put all of the fish together, we can see some important individuality in their movement sequences, as well as some shared characteristics or shared features, namely periods of rapid movement where fish are moving between different gates in quick succession, as well as times when they have protracted long periods of residency at a single gate. Sorry. And so when we overlay this then with the seasons of the ice-free and ice-covered season, we can see that high movement is primarily concentrated during the ice-free season when resources are more abundant. Looking at this then a bit more quantitatively, we can use a variety of activity metrics calculated in each season to compare sculpin in the ice-free and ice-covered seasons. So shown here is a simple roaming index that quantifies how much space available within Tremblay is being used. And we can see this significant reduction during the ice-covered season. And this pattern of reduced activity in the ice-covered season holds true regardless of the metric that you use. Looking then back at and putting this together with the feeding, a few important takeaways are that we can say, and that we have confirmed that sculpin are resident within Tremblay Sound year-round. They were detected at all times of the year. But this does not mean that they're not exploiting the resource pulse. So we saw them feeding quite heavily during that ice-free season. In addition, that their diet is flexible and changing rapidly with the available prey, as is their activity. That's reflective of that seasonal condition, that as the conditions improve or become warmer, more productive, they become more active, mobile, and far-reaching. Some implications of this, it does tell us that not only migratory species exploit resource pulses, so it's important to consider residents as relying on pulses as well. But perhaps more importantly, that residents, due to their limited mobility, may be unable to seek out alternatives as pulses change and maybe dampen with climate change. And this is important to consider because oftentimes residents, especially sculpin, make up a high level of biomass within their environments. And so this has been suggested before, but is really furthered by my research. This positions sculpin as a valuable indicator of ecosystem condition. And so it's important to keep them in mind in evaluating these ecosystems with climate change. At this point, I'm going to go ahead and stop, say thank you to everyone for listening, open myself up to any questions, but please contact me at this email address if I don't get your question now. Thank you.
All right, great job, Nathan. So as he said, we'll open it up to questions. You can either raise your hand and unmute yourself and ask the question, or we can post our questions in the chat. So I see Michael has his hand raised.
Actually, I was doing a clapping emoji. Sorry, I'll let the chat questions go.
Julia?
Yeah, so you had mentioned that for the feeding studies, the sculpin were like really bloated, like golf balls. Do you think now that you have like the existing data on relative consumption, could you use Girth to assess relative consumption?
That's a very interesting idea. I think, unfortunately, since we weren't collecting Girth measurements, we wouldn't be able to compare those now to see whether or not they are particularly linked. But I think, especially with something like sculpin that have a really unusual body form where they have big fat heads regardless of the time of year, Girth actually is going to vary just with time of year and overall development of the fish as well. So I don't know how rapidly it will change back and forth in the same way as consumption might. But I think that's an interesting thing to consider that might be less invasive than having to kill the fish to collect its stomach contents. There are other ways to collect stomach contents, but that's an interesting idea.
Liz Harvey asks, can you speculate on the impacts to the sculpin as ice-free times of year are maybe getting longer?
I can certainly do just that, of speculate. Unfortunately, the scope of this project is not quite broad enough in order to make definitive claims. But I can certainly suggest that from some additional data that we've collected, that sculpin may associate with those ice resources, and so If ice is lost earlier in the year, may last not as long, that they might have to work harder in order to find resources, and so it might become more difficult to feed at these high levels that we do see them at. That to me is the most sensitive change that sculpin might feel from ice loss, but it's very speculative.
All right, great. That's about all the time we have for questions. As a reminder, we will be opening up the breakout rooms after all 4 presenters have gone, so you guys can return to any questions that you may have had. And with that, we'll move on to the next speaker. Kai?
Thank you. Yeah, thank you, Jesse. And thank you, Nathan. Very exciting talk and very exciting field study in the area, you know, the high Arctic. You probably had a great time working out there. We're moving home to the Gulf of Maine with our next speaker, Carrie Diakins. She is a master's student in the oceanography program, and she conducted her master's work in my lab on the effects of environmental changes on heterotrophic microbial activities in the Gulf of Maine and the Northeast Pacific Ocean. And Carrie, if you're ready, please take over and tell us about your exciting research.
I will. Just doing the check. Everyone can see the screen, right? No? Yes?
Wait, we can see you right now. Yeah.
Okay, I'll try again. Share screen, share to share. Okay, that looks right.
That looks much better.
Okay.
Thank you.
Okay. Well, hello everyone. Thank you all for being here, and thank you for the opportunity to present my research. I'm really excited to share what I've done on my master's program. I'd like to first acknowledge that funding for this project is through the NSF EPSCoR grant in collaboration with other partnerships through Arizona and Maine as well. I'd also like to acknowledge UNH and the Opal Lab for providing resources and contributing to this research. It's hard to believe that over a year ago, we were shut down due to COVID-19. Suddenly, the term essential workers became a term used referring to those who were doing the jobs that are required for a functioning modern world. It's the jobs that may have been overlooked before, the jobs that don't always get the spotlight. It's the essential work that keeps important aspects of our daily life moving. As my title presents, essential workers of the sea are the organisms that are doing important and essential jobs that you may not always notice. You may not always notice them, of course, because they are unseen by the naked eye. Microscopic organisms, or microbes, work behind the scenes, keeping our oceans clean and clear, and the processes they mediate are critical to ocean ecosystems. Microbes exist throughout the entire ocean, from surface waters to the deep sea. Relatively recently, scientists discovered just how abundant microbes are in the ocean, and their role in the traditional food web was previously overlooked. In the traditional trophic food web, phytoplankton harvests the sun's energy to grow and reproduce, providing food for the entire ocean food chain as one organism feeds on another. However, this leaves out the indispensable role played by microbes. Included in this figure is the missing piece, the microbial loop, the piece where microbes are responsible for organic matter recycling, nutrient remineralization, decomposition, and carbon flow through the marine food web. One of the critical nutrient cycles microbes are involved in is the cycling of carbon. Human emissions have raised CO2 concentrations in the atmosphere, and you may be surprised to find out that the ocean absorbs up to 25% of this atmospheric carbon, and microbes help make this happen. Here, primary producers fix atmospheric CO2, releasing it as dissolved in particulate material, and it's up to the microbes to process this organic material and ultimately export carbon to the deep sea, where it can be sequestered on timescales of up to 1,000 years. Through this process, microbes ultimately determine the net CO2 removal from the atmosphere. Given that climate change is warming the ocean, changing pH, and even slowing ocean circulation, it's important to figure out if any of these environmental changes will affect microbial processes such as this. And this figure mainly represents open ocean processes, but in the coastal waters, there are even more variables involved. coastal ecosystems are subject to rapid changes of organic matter input. Carbon, nitrogen, and phosphorus, noted here as C, N, and P, are added to coastal waters through riverine input, land, runoff, storms, precipitation, and erosion, all of which is predicted to increase and/or worsen as a result of climate change. There are many interactions between air, sea, and land, and climate change threatens to upset the balance between them. Microbial activities in coastal zones and the open ocean become increasingly affected by environmental factors, such as temperature. And in the Gulf of Maine particularly, temperature is a highly relevant variable. In the Gulf of Maine, there's been extreme temperature rising compared to the rest of the global ocean, where especially over the last 10 years, the yearly average sea surface temperature has been rising, and overall, the Gulf of Maine has warmed at a higher rate than the entire global ocean. In the Gulf of Maine, the microbes have been experiencing huge seasonal fluxes in temperature that are predicted only to become more extreme. And how the rates of activity may change in the future is unknown. It's important to explore how microbes are acting under the current temperature ranges before we can predict what will happen if things get worse. Some studies have shown that higher water temperatures have a strong effect on microbial activities. In the comparatively warmer Mediterranean Sea, for example, temperature is a major driving factor, where at higher temperatures, there's greater metabolic activity. From results such as these, the authors predict that an overall increase in microbial processing will occur in a warmer ocean. In this research, I explore the relationships between environmental parameters and the activity of microbes in the Gulf of Maine. One of the topics I explored is the relationship between temperature and microbial activity. Based on the temperature variability in the Gulf of Maine, one of my main hypotheses is that microbial activities in the Gulf of Maine will follow seasonal variation in sea surface temperatures, where higher rates of activity are enabled in warmer waters. In my study, I took samples from 3 locations in the Gulf of Maine over a year, from Booth Bay Harbor, which is up at the top, Jackson Estuarine Lab, and Coastal Marine Lab, both part of UNH. You may be aware of these. I think some of you work there. I was able to capture a relatively wide range of temperatures among these sites and determine how patterns of activity change under the current conditions. Now, when I say microbial activity, I am really talking about enzymatic activity, because what microbes are doing is breaking down material, and they do this by producing enzymes. Much of the organic matter pool is dominated by high molecular weight polymers or particulate material, which are too large to pass through the cell membranes of microorganisms. In order to acquire nutrients they need, like carbon, nitrogen, or phosphorus, microbes produce specific enzymes to break down polymers outside of the cell prior to uptake. Enzymes can be attached to the outer cell wall, like the ectoenzyme in this figure on the left, and they can be released into the surrounding environment, like the orange Pac-Man shapes in this figure. These enzymes cleave the bonds within complex organic matter, releasing monomers, which can then be taken into the cell. So as this figure shows, the the Pac-Man shaped enzymes are breaking down polymers into quote unquote bite-sized pieces. And the enzymes I studied are beta-glucosidase, leucine aminopeptidase, and alkaline phosphatase, enzymes which are naturally found in seawater as they are involved in the 3 major element cycles, carbon, nitrogen, and phosphorus respectively. After evaluating the rates of enzymatic activity by BG, LAP, and AP, I found patterns that change seasonally and between sampling sites. For example, the rates of activity at Coastal Marine Lab displayed an interesting pattern. Here, a plot of AP activity on the right has hydrolysis rates on the y-axis and sampling date along the x-axis. And on the last time point in July 11th, AP performed maximum rates of hydrolysis, and not shown here, but the same trend applied for my other enzymes. So I'm interested to know if an environmental parameter is driving this activity. Did something change in the water at this time? For example, I looked into temperature. However, running a correlation to sea surface temperature tells me that temperature did not have a strong effect on enzymatic activity. In the plot on the right, these data points were more random than they were correlated. So the correlations to temperature were not strong at the Coastal Marine Lab, but were they strong anywhere else? Well, in short, not really. The relationships with temperature were weak for each enzyme across all 3 sites in the Gulf of Maine, except for one significant correlation between the activity of LAP at Jackson Lab. So why aren't we seeing the nice correlations like we see in the Mediterranean Sea? Well, the one correlation with LAP leads us to the answer. We only found this correlation at one site, which happened to be the warmest site out of our 3 coastal areas. So what I saw was in lower temperatures, temperature did not seem to have an effect on enzymatic activity, but at warmer temperatures, it began to have an effect. And this may be because enzymes function within specific temperature ranges and have specific optimal temperatures where they perform maximum rates of hydrolysis. The temperatures in the Gulf of Maine were generally too far below the optimal range in order to see an effect. Except the correlation with LAP provides evidence that at the warmest temperatures, there started to be an effect on enzymatic activity as the temperature at JEL started to reach near the optimal temperature for LAP activity. If the temperatures were to continue to rise and the Gulf of Maine loses its winters, we may start to see the same patterns that the Mediterranean Sea does. In a short summary, climate change is altering the ocean and therefore the working conditions of the essential workers. Yet it is not known how microbes will respond to future, more extreme environmental changes. In my study, I aim to address this issue by first collecting data under current climate conditions, focusing right here on the shorelines of the Gulf of Maine, where temperature is an especially relevant variable. And I found one telling correlation between enzymatic activity and temperature under the current climate conditions. So in conclusion, in the Gulf of Maine, rapidly rising sea surface temperatures threaten marine ecosystems. However, under the current climate conditions, temperature is not the main driver of microbial activity. If temperature were to significantly rise, then it would begin to affect enzymatic activity and therefore increase the rate of cycling that microbes perform. All in all, the results from the study show that under the current climate conditions, the balance of microbial systems is still intact. However, changing ocean conditions are starting to push that boundary, and repercussions of which are yet unknown. But a better understanding of how microbes interact with their current environment will only help us better predict how they will respond to change. Thank you for listening. If you have any questions, I'm happy to go more in depth about any topic. Thank you.
Excellent job, Carrie. We have a question in the chat from Liz Harvey. How do you detangle temperature impacts and likely shifting microbial population compositions over the seasons?
Microbial composition is one of those big questions. That's a very good question. Because microbial composition can change seasonally, it's likely to have an impact on the activity that's occurring. However, the scope of my study didn't measure the microbial composition. I was able to get some information on phytoplankton composition, which gives me a little bit of insight, and it was able to detangle some of the patterns we found. However, microbial composition specifically, I didn't quite include in my research, but that's a good question.
Other questions you guys can shout out or put in the chat. So David asks, are there enzymes that have cooler optima?
That's a good question too. There are lots of types of enzymes that exist in the ocean, though the 3 main ones that I studied, which were BG, LAP, and AP, their enzyme optima temperatures were all a bit higher than generally seawater gets, because a lot of them were up in the 30, 40, 80 degrees Celsius range, which is just absurd, I think, for sea temperature to get to. But I could imagine that there are other enzymes exist that do have cooler optima, which may be thriving in the temperatures we see now. Thank you.
We have enough time for one more quick question. Paul says, I wonder how the temperature effect might translate into a microbial looping effect.
Into a microbial
looping effect.
Oh, how temperature may translate into a microbial looping effect. I think that's a good thing to wonder for sure. I'm not exactly sure what you mean by microbial looping effect, but it is interesting to think about temperature affecting the types of microbes that might excel under these new conditions that climate change is given, and how perhaps there'll be a whole new composition given the warmer weather, and how that might change the types of elements that are being cycled through, and how fast carbon can be sequestered, or how fast phosphorus is being used, and how it might change the overall dynamics in the entire ecosystem. Thanks.
All right, nice job, Carrie. And with Kai, we'll move on to our next presenter.
Thank you. Yeah, nice job, Carrie. Thank you very much for your presentation. Our next speaker will be another marine biologist. It's going to be Michael Dougherty, and he is a master's student in Elizabeth Fairchild's lab, also scheduled to graduate this summer. And he's going to talk about sea lice, and we heard some interesting stories and research this morning already about sea lice, and he will be talking about sea lice incident at a trout farm in light of farm management. And before I take it over to Michael, I also want to mention that he is the president of the American Fisheries Society, UNH Student Summit, and he's very active in this role. He organized a conference last fall, and I think that's a great thing to do during your graduate student life. And now I'll take it over to Michael, and he was going to tell us about his research.
Yeah, thank you very much. So as I share my screen, I'll just make one quick correction, in that I'm no longer the president of the American Fisheries Society subunit. However, our Nathan Herman here has taken over that role, and he's doing a great job. But let me start this. And I do appreciate the introduction. And I just want to set my own timer here. So yeah, thank you. My name is Mike Dougherty, a master's student in DBS under Dr. Elizabeth Fairchild. And so I will be talking about parasitic sea lice populations in an experimental salmonid aquaculture system in New Hampshire waters, and then using lumpfish as a possible solution, which we have heard about already a little bit today. And I'll spare you some aquaculture background, but I do want to point out a couple of points. That Atlantic salmon is the most imported finfish in the United States, and essentially all of it is farmed. So it's not only a high value product, but it is also a high valued farmed product. We do not have Atlantic salmon operations here in New Hampshire, but rather we do have an experimental aquaculture station that's growing out steelhead trout, which is a very close relative of Atlantic salmon. And that's Aquafort, operated by New Hampshire Sea Grant. And both species are impacted by sea lice. So sea lice are ectoparasitic copepods that will feed on the mucus and the skin and the blood of the fish that they infect. And they are quite costly. They cost Atlantic salmon farmers and salmon farmers in general hundreds of millions of dollars each year. Chemotherapeutic treatments are commonly used to mitigate lice infestations, but there are environmental concerns to those, as we kind of heard about earlier. So folks are looking for alternative lice treatments. And as we know, one of those is cleaner fish. Cleaner fish are simply just species of fish that will clean parasites off of other fish. And they can be a more natural way to mitigate lice infestations than some chemical treatments. Specifically lumpfish, Cycloteris lumpus, which again, we've heard about already a little bit today, are quite effective at cleaning salmon. Studies show that they can actually decrease salmon or lice loads on Atlantic salmon by 60 to 100%. Something else we know about lumpfish and from recent research is that if you're using them as a cleaner fish, you do need to supply something called a hide, which is simply a structure that you'd put in a salmon pen that provides some shelter and habitat for the lumpfish. And so that brings us to our own research questions. First of all, we want to explore to what degree sea lice impacts steelhead trout, specifically in an aquaculture setting here in New Hampshire. And we know that lumpfish can reduce lice loads on Atlantic salmon, so we want to examine whether the presence of lumpfish can reduce lice loads on steelhead. And also, do different hide designs affect lumpfish cleaning behavior? So to address that first research question, we decided to do a lice assessment out at Aquafort, which is again that experimental aquaculture station operated by New Hampshire Sea Grant. And so we started this in early November, and we go out on a weekly basis, and we're actually continuing to collect data even now. But each week we go out and we take a subsample of 15 trout out of the pen, and we place them in individual freshwater bath treatments. And after about 2 minutes of exposure to freshwater, lice will start to fall off the fish. So after that time period, we just put the fish back in their pen, and then we collect the contents of those freshwater baths, and we bring them back to the lab for analysis. And we're looking at a number of metrics here. So first of all, we're just counting the number of lice per fish, which gives us our mean lice load. We're also recording the species of lice that we observe. Specifically, we're looking we're keeping our eye out for a couple of different species. One of them is L. salmonis, which is a salmonid specialist. And so they only infect salmonid fish. The other one is C. elongatus, which is more of a generalist parasite that will infect all sorts of fish across the Atlantic. We're also recording the sex of the lice, the number of gravid females per fish, which could tell us sort of a broad reproductive output. And we record the estimated life stage of the lice. So just to share some of our results, we do have sort of a variation in lice loads over the course of the season. That blue line represents our total mean lice load. And you'll see that it increased as the fall and early winter went on. And then about mid-January, it started to decline. However, last week, we did see a little uptick. That orange line represents our mean gravid females per fish, which follows a somewhat similar pattern. And as far as species go, we did not see any of that salmonid specialist L. salmonis, fortunately for our trout. We did see C. elongatus, which again is that generalist parasite. And that's most of what we've seen. However, we did observe a handful of individuals of C. curtis, which is commonly known as the cod louse. So this one right here is C. elongatus. And then this is our cod louse, C. curtis. So that brings us to our next couple of research questions. First, does the presence of lumpfish reduce lice loads on steelhead? And again, do different hide designs affect cleaning behaviors? And forgive me, I'm going to go back one. So our experimental design actually starts with this image here. This is showing 6 small experimental pens that we used for our design. We have these floating off the pier at the Marine Research Complex in Newcastle. And in each of those 6 pens, we stocked 15 trout. And then 4 out of 6, we stocked 3 lumpfish each, leaving 2 pens to act as our control. And in our 4 lumpfish pens, we tested 2 different hide designs. One of those hides is our PVC panel, which is really just a pipe cut in half and then secured within the pen. And then the other one is our fake kelp hide design, which is a length of rope interwoven with some strips of black plastic sheeting, which is supposed to mimic some sort of macroalgae. And so with this experimental design, we conducted 2 separate trials. Each trial lasted 5 weeks. And much like our sampling at Aquafort, we sample for sea lice on a weekly basis. And it's very similar. We take the fish out, we put them in their individual freshwater baths. We then collect the contents and analyze them back at the lab, looking at those metrics I described earlier. It is a bit more work because rather than doing 15 trout per week, we're now doing 90 trout. So it was definitely a team effort here. And then just to share some of our results. So I'll start with our cleaner fish treatment. And that's just pens that, looking at lice loads in pens that had lumpfish present or pens that did not, our control. And so in trial 1, we did have a week-to-week effect on lice loads, which we did not see in trial 2. However, in both trials, we did see cleaner fish treatment effect. And in both trials, we see, with the exception of 1 sampling date in each trial, there does tend to be lower lice loads in pens with lumpfish than in our control pens. And just to visualize this a bit easier, I did put together these figures, which are just the means of the lice loads for each trial. And so we'll break that down by hide design. And again, we see a week-to-week effect on lice loads when we compare hides, which we do not see in trial 2. And we do see in both trials, there is a hide treatment effect. However, in trial 1, the significant difference is between our kelp hide designs and our PVC hide designs. In trial 2, it's really just between our both hide designs and our control. So just to make that a bit clearer, there is a significantly lower lice load in our pens with kelp hide designs than our pens with PVC hide designs. And in trial 2, we don't see that significance. And to circle back to our research questions, we do see lice loads on steelhead in New Hampshire waters and in aquaculture setting, but it's mostly Caligus elongatus with a handful of Caligus curtis. Fortunately for our trout, we don't see that Salmonus specialis L. salmonus. And those populations do vary seasonally. And we can say that lice loads are reduced on steelhead when lumpfish are present in our experimental pens. And I think it would be pretty helpful to maybe do this again in maybe a larger scale pen. And as far as hide designs go, we did see in one trial a difference between kelp hide designs in that they had lower lice loads than PVC. However, in trial 2, we do lose that difference, or we don't see that. And it's important to note that there are variations in lumpfish size and trout size that have impacts on cleaning behavior, and then ambient temperature can also play a role in the lice loads. And so with that, I want to thank our funders, New Hampshire Agricultural Experiment Station, New Hampshire Sea Grant, USDA, Northeastern Regional Aquaculture Center, and special thanks to my thesis committee, the aquaculture team at New Hampshire Sea Grant, my graduate and undergraduate colleagues, and the Coastal Marine Lab supervisor, Nate Reynolds. And I will happily take some questions.
Great job, Mike. We'll start off
Thank
right
you.
in the chat. Nathan Puri says, what do you think are the biggest drivers of the increase and then decrease of lice loads through time at the aquafort? Is it temperature? Is it loading success on the steelhead, or something else?
That's a great question, because from everything I've read, it would make sense that it would be temperature. However, as temperatures cooled between November and January, that's when we saw the incline. So it's really hard to say. I definitely was not expecting to see the results that we did see. And I think some of the aquaculture team was not expecting that either. So it'll be, I think I can best answer that after the next few sampling dates as water temperatures start to warm.
Great. Diane raised her hand.
Yeah. Hi, Mike. Thanks. That's really interesting. As I understand, there's a narrow window where lumpfish are effective, and with the Gulf of Maine warming, can you comment on the use of lumpfish 10 years from now or 20 years from now, and what are the alternatives?
Yeah. Something I didn't mention is that various species of wrasse are also used as cleaner fish. And the benefit of using lumpfish is that they're very effective in cooler water. So it might be that certain wrasse species can be implemented maybe alongside lumpfish, or maybe in their place in the future. But currently lumpfish seem to be the most effective as waters sort of cool seasonally or in cooler regions.
All right, great job. As a reminder, we're going to open up the breakout rooms. So if I missed any questions in the chat, you can feel free to ask those when we move to the breakout rooms. For now, we'll move on to our last presenter.
Okay, thank you, Mike. As Jesse said, we'll move on to our final speaker, which will be Sarah Benson. Sarah is a master's student in the oceanography program, and she conducted her work in Robert Letcher's lab in Opal. And she worked also on the effects of environmental changes, especially temperature, on the microbial breakdown of organic matter in the Gulf of Maine, like Carrie was already talking about earlier. I do want to mention that Sarah was also busy doing TA work during her entire career as a grad student, and she did a fantastic job TAing, receiving the Graduate Student Teaching Award for her work in the Earth Science Department for Intro to Oceanography. So thank you very much, Sarah, for your work as a TA, and you will see that you will join me later in congratulating her in her fantastic work as a researcher as well. So please, Sarah, take it over.
Well, thank you, Kai, for that great introduction. As Kai mentioned, my name is Sarah Benson, and I am very happy to talk to you all today about my master's research of warming temperatures increase microbial consumption of marine organic matter. Before I get into my talk, I would love to acknowledge my committee and my co-authors of Dr. Robert Letcher, Dr. Kai Ziervogel, and Dr. Elizabeth Harvey. My computer response, there we go. In addition to my co-authors, I would like to thank the research vessel Gulf Challenger crew for taking me out to sea, and the UNH Water Quality Analysis Lab for running the carbon and the nitrogen data I will be presenting to you. Also, shout out to Carrie, my colleague, for her introduction to be my introduction. So we'll go ahead and get started here. So the ocean is full of life, but 97% of the biomass in the ocean is microscopic and being invisible with the naked eye, as 1 liter of seawater contains more than 1 billion bacterial cells. Marine microorganisms, or microbes, dominate the global ocean, metabolic activity, and are fundamental to the ecosystem as they recycle organic matter and provide essential nutrients to other organisms. Through microbial respiration, dissolved organic matter, notated as DOM, and particulate organic matter, notated as POM, are consumed along with oxygen and regenerated into bioavailable nutrients such as carbon dioxide, nitrate, and phosphate. Additionally, they're contributing to the biological carbon pump, where phytoplankton are absorbing atmospheric CO2 through photosynthesis, producing organic matter that will sink down to benthic organisms such as lobsters, providing them with nutrients and sequestering carbon to be preserved in the deep ocean for millennia. An effective biological carbon pump is when our primary production is outpacing our respiration, sequestering more carbon into the deep ocean. Whereas an ineffective biological carbon pump is where respiration is outpacing primary production, reintroducing carbon dioxide back into the atmosphere and reducing the amount of nutrients that make it to the benthos. From the metabolic theory of ecology from Brown et al. In 2004, photosynthesis and respiration rates should increase with increasing temperatures, which was later verified by Regarde, Dejo, and Duarte in 2012. From their study, they yielded a prediction that microbial organic matter consumption may outpace primary production in a warming ocean, creating an ineffective biological carbon pump. Now, the sensitivity of metabolic rates to temperature is often most expressed as the temperature coefficient, or the Q10, which relates the fractional change in a rate for an increase of 10 degrees C. Now, many biological reactions have a Q10 value near 2, so this green line here, meaning that with an increase of 10 degrees C, the reaction rate will double. So for example, the organic matter respiration of soil is between 1, so no temperature stimulation, and 2.6. Now is determined by Joe et al. In 2009. If a reaction has a Q10 value that is less than 1, it means there is a reverse sensitivity and the reaction will be faster in a cooler temperature. However, this sensitivity for microbial respiration of marine dissolved organic matter and particulate organic matter is relatively unknown in the ocean. Instead of focusing on the global ocean, we can focus right in our backyard in the Gulf of Maine. The Gulf of Maine is a mid-latitude basin in the northwest region of the Atlantic Ocean and has been warming 99% faster than the global ocean since 2012, as mentioned previously by Carrie. This is thought to be a result from a northward shift in the position of the Gulf Stream, which cuts off the cool waters from the Labrador Current, which flow south around the Canadian Maritime provinces and fill into the basin. It is clear that the sensitivity of marine microbial consumption of organic matter to temperature warrants further study. So with a focus on the rapidly warming Gulf of Maine and the expected ecological and biogeochemical consequences of ocean warming, this study sought to answer 3 questions. The first being, do experimentally warmer water temperatures correlate to a faster rate of organic matter consumption by the in situ microbial community? Do these warmer temperatures correlate to an overall larger amount of organic matter consumed compared to the cooler in situ temperature? And what is the Q10 of organic matter consumption in the Gulf of Maine? These questions were studied by collecting seawater during the fall 2019 and 2020 Gulf of Maine phytoplankton bloom, and locations are depicted in this top plot by these triangles. So blue triangles 2019, black triangle being 2020. Additionally, we collected coastal seawater at this red square to collect water that had a larger substrate amount due to the coastal influence. For the purpose of this presentation, I'm only going to be focusing on one of the 3 treatments that we were using to solely focus on that organic matter addition. So this treatment will be referred to as FEPOM, as 80% of the water that was collected at the triangles was filtered to remove organic matter and the in situ microbial community, with 20% of that water left as whole seawater, so an inoculum. Then with the coastal seawater, we did filter it to get that additional organic matter and we put it into this water. Once that was all mixed up, it was allocated into 1-liter bottles, then placed onto a roller table to prevent particles from settling, and then into a dark temperature-controlled incubation room to prevent any photosynthesis from occurring. At each time point, I did pull out 3 bottles from the in situ versus the experimental, and then each bottle had these samples pulled for later analysis. 2019 had an incubation duration of 8 days, with 2020 having 14 days for the experiment incubation. For this presentation, I will only be focusing on the trends we see in the particulate organic carbon and particulate organic nitrogen pool, but would be happy to discuss all of my other data with you at a later time. For the nomenclature that you will see in the preceding slides, the treatment is FEPOM, so the filtered inoculum particulate organic matter. The elemental pool, carbon is notated by C, nitrogen by N, and then the temperature that it was incubated at. So first, we'll go into the 2019 experiment. The top plot is going to be the in situ, and the bottom plot is the experimental, and this will hold true for the remainder of the plots you will see. And then for particulate organic carbon, the trends we were expecting to see is decreasing concentration with time. And for the 2019 experiment, we do see overall net consumption with a faster rate and overall larger quantity of POC consumed in the warmer incubation. For particulate organic nitrogen, again, we should see concentrations decreasing with time. And we do have an overall net consumption, again, with the faster rate and overall larger quantity in the warmer temperature incubation. Moving into our 2020 experiment, we increased that duration to now 14 days, but we did have 2 separate consumption events for our particulate organic carbon pool, with our first event being from T0 to T2, and the second being from T8 to T14. The first event, we experienced POC consumption, faster consumption in the warmer incubation and an overall larger amount consumed. But for event 2, there was a faster consumption in the cooler incubation treatment, but an overall larger amount consumed in the warmer incubation treatment. For our particulate organic nitrogen pool, we did see a delay in our consumption. So T6 for the cooler incubation and T4 for the warmer incubation. I did start the consumption at T4 to keep the treatments comparable. Before the delay was initiated, the warmer incubation did experience a faster rate of consumption. However, after the delay, the cooler incubation saw a faster consumption rate. However, the warmer incubation still had an overall larger amount of PON consumed. I did calculate the Q10 for my elemental pools for both experimental years. A reminder that the Q10 value of 2 means that the reaction rate will double with an increase of 10 degrees C. As we can see in the 2019 experiment for both carbon and nitrogen pools, we did have a Q10 value greater than 2, meaning that the carbon reaction consumption would be 266% faster with a 10-degree increase, and nitrogen being 342% faster. Now for the 2020 experiment, we do not see those numbers at all. So carbon had a Q10 value less than 1, meaning that it had a reverse sensitivity and the reaction rate was faster in the cooler temperatures. And for nitrogen, it was slightly above 1, meaning that with a 10-degree increase, the nitrogen consumption would be 121% faster. Now, the overall main conclusions from my talk today is that the warmer temperatures exhibited faster rates of consumption with an overall larger quantity of marine organic matter consumed. Temperature does play an important role in organic matter consumption, but it's not the sole factor. Longboard et al. In 2018 said that a Q10 value of 2 is not universally applicable. And as we saw in my Q10 data, there is variability that needs further investigation, with specific considerations for organic matter composition, if there's any nutrient limitations, any seasonal variability before we apply it to a global model. Now, with microbial consumption of organic matter increasing with temperatures, less organic matter would sink to our benthic organisms, reduce their food source, and risking the entire marine food chain to collapse. More areas of CO2 outgassing will likely occur across the ocean surface and reduce the amount of carbon being sequestered to the deep, and creating an ineffective biological carbon pump. All of this will further contribute to our ever-changing climate. And with that, I would like to thank you all for tuning in. And if you have any questions, to go for it.
Excellent job, Sarah. With that, any questions? So from Nathan Fury in the chat, he says, what do you think carbon's response was? Why do you think carbon's response was much more variable than the other elements?
That's a great question. And I have plenty more other data to show you if you would like to see it. But I did have cell counts that were taken into consideration for my project. And when heterotrophic bacteria consume carbon and they have a bacterial growth efficiency, if they're efficient, they will put that into their biomass instead of respiration. Instead of an ineffective bacterial growth efficiency, they would just respire it instead of growing. So I actually saw an inverse correlation with my POC consumption with my cell counts. So they were allocating all of that carbon directly into their biomass. But like I said, I have all that data I can show you and be happy to talk to you about it more.
All right, Anita asked, do you have any idea of what the microbial composition was in your experiments and whether there was a difference in microbial composition between the 2019 and 2020 runs?
So I did collect the data for that DNA composition. With the COVID-19 shutdown, I wasn't able to get to it and just kind of was out of the scope of my timeline for a master's, but I do have the sample, so it can be run for whoever comes next in my lab. It's definitely a consideration as there could be some differences in our composition to see those different trends. I know one of my colleagues before me for his undergraduate, we did see a slight shift in the composition. So I definitely don't think it's out of the question and would have loved to see kind of that data and how it correlates to the trends that we did see from the 2 different years.
All right, then we have one more from Paul. Do you think there were other limiting factors that might have affected consumption rates, for example, DO or pH?
So possibly, yes, not specifically dissolved oxygen or the pH of the water. More what we were looking at was, so another part of my thesis was looking at the elemental composition of our organic matter and if it was super carbon rich or nitrogen poor and what those nutrients are limiting towards our organisms. For dissolved oxygen, due to being in the euphotic zone and we aren't in a hypoxic area, and we did have a gap of air present in our bottle. So I don't think that they were limited by oxygen at all. But yes, there are definitely other factors that need to be considered in our consumption rates that's not solely temperature.
All right, great job, Sarah.
Thank you all.
Thank you, Sarah and Jessica and everyone else who contributed to this, the second session, and of course, the first one this morning. I see that the break rooms are now ready to be populated. If you're interested, please go and meet our presenters in the break room for some continuing discussion of their exciting research. With that, I leave it to the break room discussions. Thank you very much again.
I think so. The rooms are all closed, so we should all be here.
All right. So I hope you all had a great discussion in your separate rooms, and the team of judges met in a judging room, and we definitely had a lively and interesting discussion. And as always, yes, Sally.
Can I just mention one thing before you announce the winner?
Please.
Could we
please.
have all of the students put your videos on and everybody else shut your videos off? So that way we'll see the students up front.
Yes.
Thank you. Sorry to interrupt you.
No. Thank you. That was actually important, and that was something I should have also
So is
remembered.
that we need? I think we need all students with videos on. So
we have Alex, Carrie, Sarah, Nathan, Grant.
there we go. Who else do we need?
Who else? Do we have Jang?
Jang is there. Sarah, yeah. One, two. I think they're all there.
Great.
Okay.
So, as I said, the judges met, and great discussion, and all the talks were fantastic, as always, very exciting, and I really enjoyed this this symposium, and it was so close. It was so close to find the winner, and kind of back and forth. But in the end, we decided that Nathan Herman is going to win the award for the best presentation today. So please join me in congratulating Nathan. Great job, Nathan. But again, please, everyone, it was fantastic. It was a great job from everybody. And yeah, I think we were, again, very successful in showing the breadth of the fantastic research that is going on by our students in the School of Marine Science and Ocean Engineering at the University of New Hampshire. So thank you very much, everyone. And I think those are the last words, the final words for today. Sally is nodding. Okay. So have a great day, rest of your day, and see you later. See you soon, hopefully on campus. Bye.
Thank you, everyone.
Next year, this is in person, everyone.
Yes, please.
Yeah.
With
Diana,
drinks.
Thank you. Yes.
Are you staying online, Diana?
Yes, and treats. I'll stay online, and treats for David Burdick.
Yes. I wonder if we, Diana, I wonder if we continue this.
Yeah, why don't I just send you another link?
Yeah, I think that's
Okay,
that's
thanks.
easier. That's
Okay,
easier.
all right. Thanks,
See you in a
everybody.
minute.
Thank you for all your
Bye.
work. Bye.
Thanks, Sally.
Bye.
Bye.